Ladies and gentlemen, thank you for coming to the second Systems in Biology reading group. This is pretty fun thing that we're now going to do regularly. Before getting into the meat of the talks, just a few words on why we think this is important. So at companies like Latch, at research institutes like the ARC Institute, Modal, and so forth, what's really driving progress in the biotech industry is very simply new molecular measurement techniques that are kind of doubling every 2 years. So whether it's single cell biology, spatial transcriptomics, proteomics, there's a wide array of these measurement tools that are progressing pretty fast. Oops, get back there. And briefly, so experiments are getting really big by the numbers. Single cell, I'm giving a few examples here. You should just focus on the numbers for people that are familiar with working with file data, because...
Can you step to the right?
Oh, thank you. Yeah, just focus on the numbers for people familiar with file data, because they're kind of shocking. So a modern single cell experiment with a 10x flux kit can generate 2 to 6 terabytes per run. Modern spatial experiments are pretty ridiculous. VizGen's Merfish Ultra can yield 7 terabytes of file data per run. And deep LCMS, so liquid chromatography, mass spectrometry, and similar proteomics experiments can yield hundreds of gigabytes per run. And the interesting thing about these numbers is they're doubling every year or 2 years, depending on the assay type. So the pitch here is why study systems X biology in the age of agents? There's a lot of interesting things happening right now. Models are getting really good at stuff. It seems possible that a lot of software engineering that we're used to is going to get automated, or already has, especially basic web development and these things. So the question remains, is like what stays durable as these trends play out? Well, as file data from molecular measurement kits continues to double, in biotech R&D, the time spent in computation will eat larger proportions of cycles. So as those curves continue to increase, the tool calls from agents doing analysis work will move from days to weeks to months as you ride the curve. And then this points to what kind of engineering becomes valuable when software traditional classes of software are essentially free. Well, it's exactly the engineering tasks that extract more performance from hardware, whether it's storage devices, I/O, CPUs, GPUs, because those are going to be the bottlenecking constituents of these multi-month tool calls in bioinformatics workflows. And then broadly, I think people here might already have this intuition, but computers are becoming vehicles for real work rather than just information visualization and dashboard type devices. So storing and transforming so much literal information, especially as these curves continue to play out, that it spills into the physical world. And this work is going to become very high stakes and expensive. If you're dispatching tool calls that take weeks to months over hundreds of machines, you're not going to be vibe coding that stuff. It's very difficult. So there's kind of the broad idea. I think biology is a unique domain because we're kind of pushing the volume ahead of many other domains. You actually don't experience those numbers in many other fields of science or even in tech. So before jumping into the talks, we have some open roles at Latch and at Modal. So we're hiring for both bioinformatics and system software engineers. Hopefully Max is going to talk about some interesting stuff. If you guys like it, come find any of the Latch folks. And then Modal, much larger company, has a bunch of roles across machine learning and systems. So we're going to...
Can Latch engineers just wave their hand real quick?
Yeah, good point.
If you want to talk to someone at Latch, here's your people.
And then same with Modal. These three, cool. And then individual speakers from ARC and so forth can plug what they have. The speaker order is as follows. I'm really excited about this collection of talks today. Really samples from problems, again, across high performance bioinformatics workflows to what kind of problems you experience at storage as single cell data gets larger, to container orchestration from tools that Modal and LatchBio have built alike. Max is going to be talking about Forch, which is a utilitarian cloud container orchestrator. We've wrestled with the pain of Kubernetes for years. It has lots of issues, and he'll get into something we built to address that, to address them. Noam's going to be talking about a very high performance tool for processing single cell data, much needed. Pavan's going to be talking about a storage format for basically huge matrices of counts for single cell. Again, like as single cell data keeps on doubling, you're going to need to rethink how you store and extract counts from this stuff. Dhruv's going to be talking about state, stack, and beyond. Very excited about that talk. And then last but not least, Ben's going to be talking about how modal is used for computational biology. So passing things off to Max for the first talk. And thank you everyone for coming. Really appreciate it.
All right. This is going to be pretty quick because I only have 30 minutes and I have like 60 slides. So save your questions towards the end, and I'll make sure to leave some time, hopefully, to answer them. So this talk is about our container orchestration tool that we built in-house, the objective of which is to replace all of our use of Kubernetes internally. And I'll go over some of the issues and motivations for why we did that, because it's definitely a strange choice by most accounts. You know, don't build in-house this stuff. But I hope it's pretty well motivated. And we'll go over some of the design decisions and some of the implementation details that I think are pretty interesting. Sorry. There we go. So first, I'm going to go into some kind of world building, what we started with. And so we started with K8s because a tool that we wanted to use, which is a workflow engine called Flight, out of Lyft formerly, is built primarily around K8s. They have other backends, but this is kind of the main one. And we decided to use AWS EKS, which is the SaaS solution by AWS for Kubernetes clusters. And so we ended up with two clusters, one of which runs the core services that are the Python scripts that are basically running most of Latch, and then a very large cluster with at currently 23,000 namespaces running customer workloads, which is measured by Flight, or sorry, managed by Flight. And what you'll see is in the simple cluster, it's just basic deployments. In the Flight cluster, it's one-off pods that Flight schedules and manages, and it's all custom resources. It doesn't really have any normal deployments. So we ran into a lot of different issues, most of them kind of paper cuts, but they end up to really, really, really hurt. And the first one, and this is something people find out about very quickly, is that everything in K8s is YAML. And YAML is not only hard to edit by hand, but it is also something that you're going to have to generate a lot of. So for example, if you have a prod in a dev environment, that means you already have to duplicate everything. And so you're not going to do that by hand. You're going to be using Kustomize and Helm, which are the two tools for it, both of which are basically domain-specific languages for generating YAML. And they have a lot of quirks, and they don't have a lot of time put into making them kind of feel like real programming languages, but they are real programming languages, and it's going to hurt to use. There's an alternative called Pulumi, which builds on top of Terraform, which builds on top of their set of scripts for managing this. Pulumi is good because it lets you use a programming language, but it doesn't really solve the problem. It just kind of eases the pain a little bit because you don't have to learn DSLs. So the next thing that we ran into very quickly is the autoscaler. This is part of the cluster that manages provisioning new nodes and killing old nodes when they're no longer needed. This is a part of Kubernetes. It's in the official repository, but it's made out of components that are managed and supported by different teams. AWS has their own part, GCP has their own part, et cetera. So they all have their own quirks. It's not very consistent. And one example of how this hurts is for AWS, if you want to scale to 0 and from 0, so to and from no running instances per node group, you have to manually attach tags to an internal resource called automatic scaling groups or something like that. And there is no kind of direction for it. The SaaS solution doesn't do it for you. It just doesn't work with scale from 0. You just have to figure it out, and you have to build your own way of doing it. So there's more, which is a lot of these are going to be EKS specific because that's the tool that we used. They don't necessarily apply to K8s, but if you don't use them, then you're going to get a bunch of other problems from not using a SaaS solution. So when we use the autoscaler on the EKS, you manage your nodes using what's called node groups. And node groups are basically how you configure the autoscaler to tell it what it can provision and what resources they have, etc. And you'll find that you can only have like 150 of them. And in a lot of cases, you want to have custom labels and taints on them, like annotations that K8s uses for scheduling. And that means that you might end up with like 4 times as many node groups as you expect, because in our case, for example, we run a separate node group per AZ, for availability zone, etc. And the autoscaler isn't very smart. It doesn't really know anything about instances. It can't choose the cheapest one. It just thinks that they're all kind of the same. If it fits, it just will schedule it. And then if you dare to change the node group while something's running in it, all your nodes will die, all your workloads will get interrupted, and you will have to start over unless you're being very, very smart about it and using custom scripts to manage everything. So more paper cut issues. On EKS, the control plane is what you're paying the SaaS provider for. They're managing this Elastic server that is fully abstracted away from you, that manages the database side and all the scripts that K8s is running, et cetera. But what this means is if that thing ever locks up in a way where you can't get a shell into it through API calls, you can't touch it, because you can't SSH into the instance, because it's not yours, it's the SaaS provider's computer. And this happened to us multiple times, and the way you solve it is you beg support to please fix it and unblock your prod. And then the other problem is we don't know how this scales, because what we've heard from AWS support is that we're running a really, really big instance, and we have no idea if doubling the cluster size would make that instance crash, and if they have a bigger instance that they would give us, we just don't know. So it's kind of a time bomb in that way. One more thing is bad error messages. K8s is written in Golang, and they use a lot of Go routines. And if you want your Go routine to have a timeout, you put it in what's called a context. And if that timeout triggers, you will get this error message: context deadline exceeded. You will have no other error details because the Go routine is dead. It didn't log anything. You do not get an error message. Now, this is already terrible, but there is another problem, which is if your pod or your task dies, it will get garbage collected. And you can't look at its logs. You don't know what happened to it and what state it was last in. Now, blessing, soon K8s will hopefully have OpenTelemetry tracing. Right now it's experimental, so we didn't make use of it. But just keep that in mind if you are running K8s. More stuff, CLI tooling. It's pretty bad. They have this weird design where everything is verb noun, get pod, get deployment, etc. It sounds like you're directly typing into a REST endpoint, because you are. And then there's other issues. We're running 2 clusters, which means 2 contexts. There's no official tool to manage them. We're using a third-party tool. There's a lot of things that work like that. And then there are certain verbs that don't really make sense as distinctions. For example, guess what get does versus describe, right? Those are completely different actions you can take on the pod, for example, and they give you completely different information, unless you're willing to look at giant dumps of YAML. Here's some examples of the tooling and screenshots. If you can't see, by the way, there's a second screen over there. It might be a bit better. On this screenshot, I'm typing kube control get. And it's giving me a big error message because I forgot to tell it the noun. I just told it the verb. Now, unfortunately, it doesn't tell me the nouns that I might want. It tells me, here's a command to list every noun. They come out in alphabetical order. 90% of them you will never use. Second problem, I type get control, get pod. Everything's super good, verb, noun. No resource found. I know there's pods in this cluster, and that is because I forgot to tell it that I want a namespace that has stuff in it. And it will not go out of its way to tell you that you made that mistake. It will just assume that you will figure it out. There's nothing in the default one, so you're on your own. All right, now moving from CLI to GUI. It's also not great. There's no official one. We use app2cube. It's OK. But if you wanted to avoid CLI and just use the GUI, it doesn't exist. API issues, because we're using Flight, which does a lot of K8s API calls, because again, it manages a huge cluster with 23,000 namespaces. You'll find out that API calls are pretty slow, from 100 millis up to 5 seconds sometimes per call. Then this is a very basic REST setup. So they suffer from the N+1 problem. They suffer from the inability to batch requests. And a lot of our scripts end up going over thousands of resources. Each call takes a couple seconds. They don't really parallelize. And so you're sitting there for 2 minutes just to list every resource that you have. It can be confusing. The docs that you see on the Kubernetes website are not for the API. They're human-written docs for human consumption. You will not find, for example, every possible field that a pod spec can have. which is really important in certain cases. There's a magic link over here that you will see in slides later that points to auto-generated open API docs that tell you what's actually happening on the API level. And the official SDK, at least for Python, is really bad, untyped, no async support. We're using Lightcube at this point, but it's a partial solution. All right, almost done with these. Cluster updates are really, really bad. EKS will just make you update your cluster when it runs out of support. They will do it for you, even if you're not ready. It will kill every workload that you're using and make your cluster unusable. You have to do it, or it will explode in your face. Now, updating this is kind of hard, because Kubernetes isn't one thing. It's actually a lot of things. And you'll see that, for example, you have K8s itself. Then on AWS, you'll have AWS Node. You will have some load balancer deployments. You're going to have a network interface, a storage interface, and probably a network security level. All of those are separate containers with individual version requirements, all of which have to match. Otherwise, you will have random errors in places. And finally, just a couple mixed things. This is a cool bomb. It's the default. And if you have too many services, you will get an error from the Linux kernel saying the command line is too long, because it just concatenates text into the command line. So you have to set this to false. It defaults to true. No idea why. Probably no one wants their pod to just not work, right? We had issues with certificate provisioning, which we don't care about, but it's forced on us because K8's webhooks only work over HTTPS despite being on a local network. There's a bunch of concepts like QoS classes or how resource limits and requests differ, which are intimately connected. QoS is determined by how the difference between those two operates that impact random parts of your cluster. For example, QoS classes influence how out-of-memory pods get collected or killed. One thing you'll see in GUIs is node shells. Those are basically crucial for debugging, but K8 doesn't tell you that they exist or bless them. You just kind of have to get this knowledge out of the community. We had issues with finalizers. This is a magic part of the resource abstraction that runs when the resource dies. It can block the resource from dying. It can crash your control plane, which is how we found out that if it does, you can't get a shell into it, etc. And then NVIDIA config is just a thing you have to figure out, and the version has to match exactly. And config map updates is something we never got to work. It kind of injects a bunch of symlinks in this weird mounted folder in there. And if you try to use inotify on it, it just doesn't work, etc. Anyway, this is life in Kubernetes land. So that's paper cuts. And you might say, well, just deal with it. It's operational complexity. It works, right? Well, let's consider some things that are actually preventing us from doing cool stuff, right? That are fundamental limitations. One of them is instance type selection. As I mentioned, node groups aren't great at this. There's a limited amount, and it doesn't consider cost and stuff like that. Ideally, we would just want to tell it, here's all of AWS. This is all that they have. Choose the cheapest one for this workload. We don't care what it is beyond that. Now, the next thing is we want this thing to run in multiple regions and multiple clouds. The reason for that is AWS had a couple periods where waiting on a GPU could take a day or more in a specific EZ, while the neighboring EZ or a neighboring region would have them, or GCP would have them, etc. And then this also opens up more instance types and more availability overall, so we can optimize costs better. Then finally, the question of bring your own compute. This is important for people with startup credits on AWS and stuff like that, and on-prem solutions for people with very strict data provenance requirements. Now, before we commit to it, we should actually theoretically consider replacing Kubernetes, right? We know that it doesn't support these future needs, but maybe there's workarounds. We know that it's annoying to operate, but we can still do it, right? What we should consider is how hard would it be to replace? And the answer to that is, well, you kind of have to build a house because there's no thing that's equivalent to K8s that isn't K8s. There's other flavors of it, different SaaS solutions, different kind of forks of it, like Red Hat has one as far as I remember. And other people are running either on serverless, like Lambda and stuff like that, or they're running on SaaS solutions, like Firebase, whatever, or they're running VMs and managing them through Ansible and Chef and similar tools. So let's consider what it would take to actually replace Kubernetes now that we know that we would have to write from scratch. Right? What does it even do? Well, if you're trying to be super reductionist about it, what it does is it Docker runs some stuff somewhere, right? It's kind of like very simple But we can split it down further. Yes, it does Docker run, but it also does other things. It auto-scales for us, keeps track of running tasks and what happened to them, statuses, logs, exit codes, resources, etc. It deals with networking. It deals with storage, persistent devices, distributed file systems. It deals with config and secrets for these tasks, which are usually injected as files or environment variables. And it has APIs, so we can take our workflow orchestrator, which knows kind of what to run after what, and feed its output into K8s to have it actually run. So, OK, seems like we're considering rewriting K8s. What would our other goals be? Well, one is we have all this trauma from context deadline exceeded. It's terrible. So we want a lot of observability, like really overkill. So one thing we're going to do is we're going to take OpenTelemetry tracing as a basic requirement. Second thing we're going to do is take that issue of disappearing pods with their errors, and we're going to solve that. We're going to say, all the state that ever changes, we're going to keep in the database so you can go and look at it later. Now, one other thing, and this is related to the idea of multi-cloud, multi-region, on-prem, BYOC, et cetera, is compute abstraction. So we want to not have to deal with anything, any of the differences between AWS and GCP. We just want to say, here's a computer, run some stuff on it, and it should just figure out how to get that done. So when we start designing something, you want to start by looking at prior work. So in our case, prior work obviously is K8s. So what works about it? Well, the first thing, and this is kind of the core of their design, I would say, is the declarative approach to everything. You never tell it, OK, here, run this container. You tell it, I want this container running. And that's a very crucial difference, and is going to lead to a bunch of very useful properties. Now, the next thing is using containers as the basic unit of work. So this is opposing to VMs or shell scripts or something like that, simple dependency management, et cetera. Containers won the world. I don't have to justify it, really. Final thing is, K8s is built around the database. You're just staring at the database. You're not really concerned about the services as much. And this is actually very good. Now, we figured that out mostly through our other products and stuff like that, where we just put everything in a database as well. Databases are awesome, and we want to keep doing that. And in our case, the database will be the almighty Postgres. So let's start designing. Postgres, because it's great. Python, because we don't care about performance, and because it's actually quite easy to write and support, et cetera. And you get stack traces instead of context deadline exceeded. Why don't we care about performance? Well, because we're going to mostly be doing API calls to Docker, to AWS, et cetera. AWS API call latency is like 100 milliseconds. Docker gets pretty slow as well. It really doesn't matter if Python takes 10 milliseconds to make this request over C that would take 200 milliseconds, right? Like, this is a completely non-consequential change. Now, let's think about what goes in the Postgres. Fundamentally, we have tasks, we have nodes, and the tasks go on nodes. So those are our tables. So one final thing with the database is we have to decide who actually gets to make rows in these tables, right? This is kind of core to designing stuff, because we want it to be secure. We can't just kind of throw stuff around. If you think about it, creating tasks is kind of safe. There's going to be users. They're going to be making tasks. Just making a task doesn't mean it's going to run or anything like that, so it's OK. Same for nodes. We'll just let a computer say, I want to be in the cluster. OK, that's fine. I mean, other than spamming the endpoint, nothing can really go wrong. Where things do go wrong is on assigning tasks to nodes. For two reasons. If a task runs on a bad node, one, the task will waste the node's compute time, which the node operator isn't going to appreciate. Two is the node will see the task's RAM, its resources, et cetera, and can steal all the data. So we can't just let people assign their tasks to random nodes, and we can't let nodes pick up random tasks. This has to be a privileged thing that happens inside the system with access controls. So let's think now about what happens in terms of the database when we say a task gets assigned. Remember that we want to have maximum observability. The litmus test for this is going to be, say we have a crash, and it happened 2 days ago, but we just noticed, right? Somebody complained, or a downstream thing crashed. How do we figure out what happened? Well, we want to be able to go and make a query against the database and get basically the full state of the system of 2 days ago. Now, the solution is three-part. One is tracing. Traces are going to be around for a while. We can go look at them. Two is table rows are going to be immutable, because if we have tasks and they hold state in the row, and then the state changes and the row changes, there's no way to go back. It's lost forever. Now, the third thing is, so how do we save state? We're going to save the state transitions in a big table. This table is going to be called something like task events. The events are going to have types. They're going to have timestamps, et cetera. That's a sort of design that's sometimes called event sourcing in kind of like really old literature. But the idea is basically when the task gets assigned, the row doesn't say, OK, my node is 1, 2, 3. Instead, a different row gets read into a table called task events that says node assigned. The node ID is 1, 2, 3. The task ID is 4, 5, 6. OK, so let's just pretend that we figured everything out except the last step. We have our task, we have our node, they're assigned to each other, great. How do we figure out what is going to happen next? Well, we'll take a Python service. We're going to call it fork node. This thing is going to run on every node, and it's going to be doing the actual Docker execing of things. So its functions are super trivial. It takes the tasks that it's supposed to take, it Docker runs them, and then when the task finishes, it writes the stuff back to the database so we know if it crashed or if it succeeded. Now, we get into some interesting implementation details here. The same three-step process in pseudocode: find, run, assign, status, right? Well, we're going to assume, like all good distributed system designers, that our stuff crashes all the time in the worst possible spot, and we have to deal with it, and there's a billion of them running, and they're all trying to kill each other. So two problems I see here. One is 2 and 3 are not an atomic action, so you can run a container and then crash. And so now, is that fine, right? Well, that's the question. So the next issue is 1 and 2. OK, we got the task that's assigned to us, but what if somebody else unassigned it before we ran it, right? Well, so let's look at it. If you crash between 2 and 3, you end up with a duplicate container. You make a container, you crash, then you restart. You come back to 2, and you make another container, and then if you crash before 3 again, you will do that again. And you'll just keep spawning containers until finally 2 and 3 happen together. Now, for the other question, it will just run on the wrong node. If we get a task that's assigned to us at time 1, but then by the time we run it, it's unassigned from us, we are going to still run it, right? Because we don't do a second check or anything like that. And so this node that it was once parent to this task, but isn't anymore, will run it anyway, which is bad. So the solutions are kind of trivial, but they're important. We're going to hold an exclusive lock over the task so that nobody can take it away from us while we're trying to run it. In a lot of cases, you would say we need a log manager. This is a distributed service we have to run. And we're just going to say, OK, actually, Postgres can just do that. And we're going to use row locks instead. So we're doing select for updates. In terms of K8s, for example, they use etcd for this, which is a proper distributed locking store. But we just don't really care about distributing this cluster. And then the next thing, before we modify a resource, we're actually going to check if this action was already completed. So before we run a container, we're going to go and look for the containers that represent the same task. And if they do, if we find one, we're going to realize, oh, somebody already ran it. Probably us before we crashed. So we're just going to recover from that instead. And this relies on good locking as well, because otherwise it's invalid. One more thing to mention is that instead of doing this part, some APIs will let you give them an idempotency key. So if you're giving the same key twice, it will only do the action once, but give you the same result both times. In those cases, you will usually save the key in the database first before you do the action, so that the next time you'll have the same key and it will all resolve properly. So here's the same code as before, but complicated to work around these distributed issues. One is this context manager in Python is showing you where the transaction is. At the end of the transaction, we'll commit, and we'll release any logs. Then in the find query, I added the for update, so that we're holding an exclusive lock. And then this is the part that does the crash recovery. Before we do running, we will check if the container exists. If it does, we're going to take its status and put it in the database and do nothing else, because it was already running, so there's no point to run it again. The reason we take the status here is it might not only have been already running, it might have been running and crashed already. So we have to recover from that as well. Otherwise, this is the clean slate. We'll run the container, and we'll record its status. Now, cool, we figured out how to get to Docker run from a correct DB state. How do we get to the correct DB state? How do we decide how to put a task on a node, right? For that, we're going to make our aforementioned internal privileged service, and we're going to call it Fork Orchestrator. And this is the thing that's going to decide which task runs on which node. There's only going to be one per cluster, but we're still going to build everything as if there's a million of them and they're trying to kill each other. So what are its functions, right? The fork node has 3. This thing has 3 as well. It has to assign tasks. Eventually, it'll have to deal with auto-scaling. Eventually, it will have to deal with resources. And the reason it has to deal with resources is because it's a very highly privileged operation. You know, like if I can attach an EBS volume to something, I can read it. So if you give me access to attaching volumes, you're basically giving me access to their contents. So whichever service is going to be doing that needs to be highly trusted. All right, and another implementation detail. If you think about it, you'll realize that containers crash, nodes get preempted, cloud resources can get changed by a person going into the cloud console and doing something. And that all means that we have to poll for status updates to make sure that our database doesn't desync. But since we're polling anyway, we might as well forget about that last part from the node script where we're writing the task status, and instead just let the next run around do the polling and updating of the status. The next slide has some code to walk over that. But the point of doing that is that we don't actually rely anymore on the atomicity between 2 and 3. Instead, we rely on the relationship between the status poll function and the function that's doing the resource change. And this gives us free, quote unquote, crash recovery because we don't have to think about it. We just have to think about this other property. And here's some code. This is how we would write it normally for auto-scaling, say, right? We would make the node, and then we would save the status. But again, we're going to have to poll for the status anyway. So here's my polling function, right? Well, the observation here is that this line with the update node status is actually completely useless now. Because when we run auto-scale and it just makes a bunch of nodes, the next time we run polling, they'll have the correct status anyway. So we can just simplify this by cutting this out. And then that makes the errors that might occur in the relationship between these two functions that would only show up in crash recovery when we don't record the status of a launched node appear every time if they're there, and forces us to have this relationship correct. So we're turning a rare crash recovery bug into a constant bug if it was there, and forcing ourselves to write this properly instead. So one more thing to mention about how to write these services is that you want to write them as kind of independent subroutines. This is following from the crash recovery and the highly distributed version of this. You're assuming that there's lots of these services, and you're assuming that they're in random parts of the program. So basically, if you're looking at one singular code base for them, what you need to assume is that you can only communicate state changes and between services, et cetera, through the database, because that's your only persistent storage. Everything else is temporary and viable to go away at any time through a crash. You have to assume that your subroutines are running in any order, and you have to assume that they can run in parallel. The benefits of this are quite numerous. It's hard to build for, but you get a bunch of things kind of for free. You get crash recovery, as we just discussed. You get the log of state transitions, because everything important has to be in the database, because we're assuming that everything in memory is completely ephemeral. It just goes away every time at the worst possible moment. So if it's important, it's in the DB, which means that when I'm debugging this 2 days later, right, everything important is guaranteed to be in the DB. There's no way for it to be designed any other way. We get scalability, because we designed everything to run in parallel and try to kill each other, so we can just make more of it if it starts running out of performance. And as I mentioned, it is harder to write. But then when you are making these subroutines, you have so many conditions that it's actually pretty easy to write the code. You're working in an environment that's highly constrained. And this is kind of what it looks like. The main function for the orchestrator, say, is going to look like a while true loop, basically just calling these things in order. But it doesn't matter what the order is. We can rearrange this in any order. So, but you see, they don't communicate with each other in any way. There's nothing passing between them, because the only way they communicate is through the database, which they can read and write independently. That's kind of a joke, but to advertise this, we just took over a Swiss village and we paid their transit depot to put our logo and name on every single tram. It's definitely not because this is an industrial village. Anyway, now we have this orchestrator, it works. How do we put it in production, right? Well, the worst thing you could do is just kind of keep adding to it and then hope that whatever comes out at the other end is going to be useful. So instead, we're going to take one of our products called LatchPods, and we're going to set a goal, put this on fork in production. Now, the things that are actually missing for that is volumes, networking, logs, metrics, tracing, public IP listening, etc. There's still a lot of work left. But overall, all it took was 9 months, and we got LatchPods to run on prod on fork. And you'll find that actually the main issues we've had were not related to designing or rebuilding a big part of the system, but rather to dealing with cloud API quirks. There's a bunch, but mainly, like if you pass a none to an AWS API for EBS volume listing, it gives you all the volumes instead of herring out, which was a problem when we started killing all of them. And the other problem is we're not big experts on Linux networking internals, NF tables, IP stuff, etc., which all really comes out as super important when you're trying to do NAT and you're trying to do firewalls, because these tasks can't just talk to each other because that leaks data, etc. So this is primarily where we get slowed down. And that's it. Huh?
We don't have a guest mic, so just.
Oh, raise your hand and I'll call you.
Why did you decide to store the initial state plus transitions to the final state instead of storing the final state and reverse state transitions back to the initial state?
The question is, when we're storing state transitions, why do we start at the initial state and go forward in time versus storing just the current state and undo logging all the changes that happened over time? I think the answer is it's simpler. I would say that it is definitely slower because with current state, you would get like the current state kind of cached. Instead, we have to walk the entire log every time for every resource. We decided that, or kind of like, if we ever ran into performance issues, writing a bunch of triggers in Postgres to keep up to date a cached version of this wouldn't be too hard. So that's why we went with it. I think this overall lets you look at the database and kind of figure out what happened more easily than going backwards, but I'm not sure. I would guess that this is more or less like equivalent in terms of complexity.
Curious, how does it compare to some existing tools? Indexable supports similar functions? This tools?
What did you mention? Which specific one? Nextflow, yeah. The question is, how do we compare to similar tools such as Nextflow? So this is really simple. I don't know if you noticed, but like this just goes from task node, run it, right? Resource management, maybe. Nextflow is really complicated, and this is kind of where Flight comes in as well. And we actually support Nextflow as a front end to this. Like we're not trying to fix the problem of what to run and when. We're just saying, here's a thing that needs to run, and then we're figuring everything after that, right? So it's like a pretty neat separation into workflow scheduling and then container orchestration. And you'll see it because like Nextflow has these plugins you can write basically, right, that do the actual scheduling, and they have one for AWS Batch, for K8, and now they have one for Fork, and that's where we fit in.
What's the biggest stress test you put it under in production?
The question is, what's the biggest stress test? Well, we put our entire LatchPod stack on it. That includes plots as well, because that's the infra is exactly the same. So that turns into what? Like a total of like a couple tens of thousands of workloads. They're not all running at the same time, but we're definitely up to like a little over 1,000 at the same time, I think, in like the peak periods. In terms of stressing it generally, like it'll get slow eventually. Most of the stuff that gets slow is authentication because we abuse RLS, but whatever, that has workarounds. In terms of like stressing the design, it just kind of works. I think we just made it so simple that it's hard to mess up very bad. Anyone else?
I actually have a second question. Do you have any concerns about the maintainability of Python compared to something like OCaml?
Question is, do we have concerns of maintainability of Python compared to OCaml? Interesting choice. I guess I don't really care as long as it's not Golang or Rust or something that doesn't have tracebacks by default, because those are essential. And then again, we built it to really overkill on observability everywhere. Yeah, I don't know. I haven't wrote much in OCaml, or like I only have some Haskell experience. Python, I think, is just generally kind of simpler to hack in, but I don't know, we'll see. Python with a lot of static typing is honestly not far behind functional languages in a lot of cases. And I think that's all we have time for, but find me afterwards if you have more questions.
Thank you, Max. Next, we're going to hear from, thank you. Next, we're going to hear from Noam. You able to share screen? We're good. He's going to be talking about a faster version of a bioinformatics workflow that has been a staple in the community called CellRanger, and how we use ideas and systems to make it faster. Thank you.
Hey, everybody. I'm Noam. I'm a bioinformatics scientist at Arc Institute, and I'll be talking today about Cyto, which is a tool I built for ultra high throughput processing of 10x flex single cell sequencing data. So I'm going to give like some background on what single cell sequencing data is, what this like modality actually looks like, because I think it's really useful to understand the context that we're building this in. So most single cell sequencing data is done via 10x, which is a single cell sequencing company. They provide a product, and it's based off of a droplet sequencing method. So basically, you have a microfluidics device that passes individual oil droplets and cells into each other. And then you try to encapsulate a single cell per droplet. In that droplet is a barcode, is a bead that has a bunch of like oligos that like have all the same barcode together. And this barcode basically sticks to the 3' or the 5' end of an mRNA. And then you basically lyse the cells, take all of the mRNA there, and every molecule is essentially decorated with a small little barcode sequence that tells you what droplet it came from originally. Here, I'll move over here. So every sequence essentially has a barcode that tells you what cell it came from originally. The throughput of this is about 10,000 to 20,000 cells per reaction per microfluidics well. And this has been kind of like the staple tool that's being used in single cell sequencing for almost 10 years now. Recently, 10x has introduced this 10x flex workflow, which is similar, except instead of capturing the mRNA directly, it actually uses an oligo pool. So you have a bunch of sequences that essentially bind onto the RNA itself. And instead of reading out the mRNA directly, you read out these probes instead. So it's a targeted list. The benefit of this, though, is that you can add an extra barcode on each one of these probes themselves. So in the first V1 of Flex, there were 16 different probe barcodes that you could use, which are called the hybridizing pool. And what this lets you do is, instead of having only a single cell per oil droplet, you can actually put a whole bunch of cells in that oil droplet. And then bioinformatically, you can separate out what every individual cell was from the hybridizing probe that was there. So the way to kind of think of this more geometrically is you have your droplet barcode that tells you what droplet it comes from, and then you have your probe barcode that tells you what probe was being used to demultiplex it. So this was put out by 10x, and then at Arc we spent a lot of time actually just generalizing this workflow to Perturb-seq as well, and to get massive throughput at that scale as well. So the only tool to analyze this type of data is actually CellRanger, which is a solution put out by 10x. CellRanger does a lot of things well. I think it struggles once it actually hits the scale that we're building at Arc. And a lot of that has to do with like three kind of like main things. One, it's not very modular. So if there are sections of the code that you want to run in isolation, you can't. You kind of have to run the entire thing. And then if there are parts of it that you don't like, you kind of have to overwrite those sections post hoc. So this is not like a huge problem for a lot of people, but when you're doing a lot of experimental design where you have non-standard regeometries or new experiments and stuff, you basically have to hack around in Cell Ranger and you have like 10 different versions of it, and it becomes very difficult to actually build. And then of course it doesn't allow additional algorithms for core components. If you don't like how they do guide assignment, well, you're stuck with it. The other thing that is difficult with it is that it has fairly poor I/O. It requires a lot of additional disk while it's running. This can be roughly 3x the input size. So if you already have a terabyte of input, while this is running, you need an extra 3 terabytes. So total of 4 terabytes total. All of that is temp files, and then at the end, you're left with your terabyte of input. This also really spikes toll, it's a really heavy toll on network file systems because this ends up making like hundreds of thousands of very small files, and this like will make your Linux admin scream. As a result of this, it's fairly low throughput. Individual runs can easily take days, and the resource requirements can sometimes be non-deterministic and just lead to crashes like 12 to 24 hours into a run, and you're like, well, there goes that time, right? I have to start that over again, but give this an extra like half a terabyte of RAM. So the requirements can be really hefty, and it just leads to just problems everywhere as you start scaling. And we are scaling. So traditional experiments are like at to hundreds of thousands of cells. I say traditional, even that's still pretty new, but hundreds of thousands of cells with fairly low depth is at the billion read regime. When you start getting to millions of cells, which is like really production scale experiments that we're doing fairly regularly now, at high sequencing depth, we're talking like multi-trillions of reads. So things that were built originally for the maybe a couple 100 million range, which was now being used at the billion range, it needs to scale up to trillions. So the costs and the turnarounds are just very high as a result. And so how do we scale to trillions of reads? The core requirements that we wanted to set out with Cyto was basically take a look at like what Cell Ranger is actually doing, and then ask the question of, can we do this faster than Cell Ranger? And can we use less resources than Cell Ranger? So we can always throw more money at the problem, just spin it up like on very expensive, just cloud instances and just pay the cost. Or let's just make it like to make it faster, or we can actually bring down the resources required and then do it faster for cheaper. So what does Cell Ranger do? It takes input sequences, it maps it to a probe library of like expected elements, it demultiplexes the probe barcodes and what I described earlier off of that decorator on the read, quantifies UMI abundances, which is like your unique molecules, and then it creates these cell by gene matrices, which are then used in downstream applications for single cell processing. So this is what led me to develop Cyto, which is a tool that we put out maybe like a month and a half ago. And Cyto is a fully modular tool that does all of these individual components, but separates them out into smaller command line utilities that you can just kind of put together. The way that it works is you take an input sample. This is a collection of sequences, like a really large input sequence here. You give it a target list, what you're mapping against. These are basically your reference sequences that you expect, and then your probe set. This is what you're demultiplexing on. You pass it into the mapper, and it creates out a bunch of smaller IBU files. These are intermediary files. These are small binary files that describe the cell and the molecule and like what it's targeting. And I'll describe that in a little bit more detail soon. But very bare bones, minimal intermediary file that's a binary file. Then we do barcode correction on the droplet to capture like errors and sequencing, UMI correction as well for the same reason, and then eventually do UMI deduplication and counting to build the cell by gene matrices that are used in single cell. There are some additional like statistical algorithms that we add on top of this to filter out the gene expression matrices and to do guide assignments, but I won't talk about that work here today. So at its core, Cyto is a mapper for abstract read geometries. And what I mean by that is that we have a collection of sequence elements that we care about. We know that we have a cell barcode. We know that we have CRISPR protospacers. We know that we have a flex probe. All we need to do is tell Cyto where those elements are in the read as just like a geometrical spec. And it handles the library lookups. It handles the mapping as a function of what you tell it the areas are. So this kind of solves that issue that we were running into, developing new different experimental techniques, but like ending up with different sequence geometries that couldn't be handled by CellRanger. So this just gives you a simple DSL or domain-specific language to tell like what the spec of this experiment is. It then looks up what that element is in our library, and then demultiplexes out into these smaller binary formats that act as intermediary steps. So this is a very simple kind of overview of like the mapping algorithm that's being used. Inside the mapper is a tool or a library that I built called Ccache, which does generalized unambiguous mismatch lookups. So that sounds complicated. What it is really is you have a library of expected sequence elements, and you want to look to see whether your subsequence exists in that library. We all know, or if you work in sequencing, you know that like sequences have errors. Like your sequencer will have a specific amount of errors in them. So if you have like a single mismatch on like your expected library, you don't want to have to like throw that read away just because you're basically one base pair off. However, doing a Hamming distance calculation for 1 trillion reads can get very expensive. So a way around that is to basically make use of the fact that lookup in a libraries or lookup in a hash map is essentially O1. And what I realized is that generating every mismatch is actually not very many sequences overall. So the total number of additional sequences that you need to store is 4, which is the amount of nucleotides, plus or times m times n, where m is the sequence length and n is the total number of sequences. So practically, if you have a gene expression library of 60,000 elements, which is what we have in our single cell work, with 49 base pairs per element, you need about 11 million sequences total to store, which seems like a lot until you calculate out that that's about half a gig of RAM, and that's nothing. So you can like absolutely keep this as just a simple hash set, look up for every sequence, whether it exists or not, and you end up getting just very cheap lookups as a result.
Why 4 and not 3?
Because you have to capture ends, which are ambiguous sequences.
Oh, I see. Okay.
Yeah. Additionally, Ccache is, so this is the upper bound, 4 times m times n. Another thing that's important is you never want to keep a sequence that has a mutated sequence that has 2 parents, right? So if you have sequences that are 1 Hamming distance off from each other, if you create a bunch of errors, you may have like a mutated element that maps to 2 parents. So you want to remove all of those as well. So this kind of deals with that that problem. I've had to build this actually for a lot of different tools. So this was originally just in Cyto, but then I just put it out into a separate library because this is actually just pretty useful in bioinformatics in general. So you can take a look at it if you're interested. The IBU format is this really simple, bare bones binary format for these intermediary steps. Basically, it is a collection of records, of fixed size records. Every one of them is 24 bytes. And they tell you what the cell barcode is, what the droplet is, the UMI, so the unique molecular identifier that kind of disambiguates sequences that are the same but are just sequencing duplicates, and the index. The index is basically what library element it is. If you have 60,000 total elements, the index just tells you which of the 60,000 it is. Importantly, the barcode and the UMI are represented as integers using 2-bit encoding. Which I've talked about in the last time I gave this talk like this, but basically is a way to kind of compress sequences which are like one byte per character into 2 bits per character, since we only have ACGT, right? So we actually only need 2 bits to represent it. So you can basically pack a lot more sequences into a single byte, and you can represent sequences as just integers themselves. So using this format for barcode correction, UMI correction, and counting, we have really cheap ways to basically read these in, and we can turn these operations, which were generally done in memory, for like Cell Ranger into streamable operations that we can run over these IBU formats. So those were kind of like algorithmic improvements to Cell Ranger, but doesn't really solve what I think is the core problem of Cell Ranger, which is this I/O problem that that we're running into a lot. And what I realized and why Cell Ranger takes up so much disk space in the first place is because it does an on-disk map-reduce pattern. So what this means is it takes its input FASTQ, which is this very, very large FASTQ, and it basically splits it up into a bunch of very, very small FASTQs. This is why it could generate like tens of thousands of very small kind of temp files. It's doing exactly this. The first step it runs is just split up that FASTQ into a bunch of very small FASTQs. And then it runs a mapping algorithm on each one of those FASTQs and then like aggregates together afterwards. So this is like a pretty common map-reduce pattern, but they're doing it on disk as opposed to in memory or via stream. And it's very clear why they're doing this. They're paying a very massive upfront cost for parallel processing. And to me, it's like, I understand why they do it, but to answer like how to fix a problem like this, it's actually a very fundamental problem. And it comes back to the fact that gzip FASTQ it kind of forces you into this paradigm. It leads to single producer multi-consumer topologies, like parallel topologies. And the reason why is that FASTQ records don't have fixed sizes, and they're actually very difficult to parse in parallel, especially once you add gzip on top, which has single-threaded decompression. So basically, you have a single producer thread that reads over these FASTQ files, and then you have consumer tasks or consumer threads that actually do like some tasks, like alignment or mapping or whatever. This works well when the consumption task is much heavier than the production task, right? If it takes a lot of time to align every single batch of records, then you can make use of your parallel architecture well. As the consumption tasks becomes really cheap, you end up with a round robin, where the producer hands off a batch of records to a consumer gets through it super quickly, and then just waits and polls and just waits until the production task comes again. So as we bring algorithms down on the consumption side, we end up running into this problem a lot more. And so the solution to a problem like this is binseq, which is a file format that I put together that's a replacement for FASTQ. And I'm not going to talk about it too much in detail here, but it was actually developed for Cyto originally. And what binseq kind of solves is this native parallelization. So it solves the single producer multi-consumer task. What it does is it captures or it puts records into these blocks on disk. And then it has an index at the end of the file that tells you where the start and stop sites are of every one of the blocks. So every thread can essentially count how many blocks are we know how many blocks are total in the file. Every thread can essentially pull however many blocks it needs to operate, decompress in thread, and you can just get native parallelization that way. I won't spend a lot of time on this, but if you're interested, come talk to me about this. So basically, taking that native parallelization task, we can solve the I/O problem by essentially skipping that on-disk map reduce and just operating on the sequences themselves via binseq files. So in summary, the architectural improvements and changes that we made over Cell Ranger is we have this native parallelization with binseq. We have this like O1 lookup with seek hash with mismatch tolerance. There's streamable algorithms with IBU with really cheap kind of like I/O there. Everything is modular. The DSL for the geometry specification is open and easy to use. And then we end up with a highly scalable and parallelizable workflow as a result. So how does it look? These are the performance statistics that we see in terms of just like wall time on a single data set that we observe. We see like a 16x improvement. So this is going from 4 hours on a single run to 13 minutes. The amount of RAM usage is about 2.4x less, and that can be brought down even further. And the part that really matters is the 30x reduction in CPU hours, because that's what you're really paying for when you like move to like a cloud computing environment. This is going from 138 hours to 4 hours of CPU hours, which makes a very big difference and actually saves Arc a lot of money. There's also a 4x reduction in total I/O. So we're not adding any more additional disk requirements over the very expensive sequencing costs to begin with. And the downstream results don't change either, which is also very important when you're building tools like this. The mean concordance we see is above 99.8%, and this is per individual cell transcript UMI counts. There are some very small fluctuations in counts because Cell Ranger filters out reads sometimes, and it's not very clear why in every case. And I've tracked down a lot of them, but not every one. So there's very, very small differences, but the downstream representations don't change, and the downstream results don't change, which is what really matters. So in conclusion, Cyto is an ultra-high throughput tool for processing 10x flex single cell data. It's a drop-in replacement for Cell Ranger. In this case, with 30x more efficiency with no loss to accuracy. In the process, we built out this really next-gen file format, binseq for sequencing data that's used in this, but also in other tools, and everything is already reliably run efficiently at production at Arc. So if you're interested in Cyto or binseq, you can read the preprints we have for them, or just come talk to me afterwards. Thank you.
We talk about a lot of components. Which component do you think played the biggest role in this?
Yeah, you ask what component played the biggest role in the speed up. I think swapping out to binseq was a really massive throughput gain. Mapping is probably the largest cost that Cell Ranger pays, and the sharding that it does at the beginning makes up a lot of the runtime for it. Yeah, so that's probably one of the biggest.
What is the time or compute cost of converting a FASTQ file to a binseq file? People have been kind of unhappy with FASTQ profiles. So do you feel like this is a generalizable thing that more workloads can switch to? What are some of the trade-offs?
Yeah, I'd say so to answer your first question, the time it takes to convert FASTQ to binseq is the time it takes to G unzip it. That's it, really. There's no additional overhead on that. In terms of the biggest hurdle with swapping to binseq is that binseq is very new, and the entire bioinformatics ecosystem is built around FASTQ. So one of the things I built with binseq is actually a way to convert from binseq to FASTQ via named pipes. So you don't actually ever need to write out FASTQs. You can make these kind of shared in-memory processes that can be passed between different tools. So that gives you a little bit more of a seamless approach. But I do think it's a very generalizable solution in that I think actually almost a lot of bioinformatics problems are I/O problems. So I do think it could solve a lot of that.
Is Cyto specific for flex nesting, or is it
Yeah. So the question is whether Cyto is just for flex or for 3' and 5' and the rest of the. It's just for flex for now. I think at some point we're planning on making it 3' and 5' as well.
Not very familiar with compute cost releases. How much is Arc actually saving for 30 years?
The question is how much did we save? In the last quarter, I think it was about $75,000 at GCP.
Those are pretty insane performance graphs. Yeah, I mean, not much to talk about after just looking at those numbers, the kind of speedups you can get. Next, we have Pavan, who's going to talk about new I/O formats for single cell. So riffing on that point, if anyone has been working on modern problems and trying to load massive amounts of single cell data, either to train models or just do bioinformatics, huge problem that is urgent today and is going to grow more urgent as these experiments scale up.
Awesome. Thanks so much for having me. It's great to be here. I really like the sequence in which the talks are structured too, because where SLAF picks up is kind of where Cyto ends. So that's really great. SLAF stands for Sparse Lazy Array Format, and I'll get a little bit into what sparse and lazy mean here. But it's a open source project on GitHub with documentation and a few example data sets on Hugging Face. So to introduce or reestablish, I guess, the scale problems that we're up against. This is a figure from Tahoe Therapeutics, and we're seeing how about a decade, a little over a decade, we've gone from single-digit number of cells to like 100 million cells, and anticipating 1 billion cells in single-cell transcriptomics. So that scale escape velocity leads to scale problems in traditional bioinformatics workflows, the kinds of things like you do like cell and gene filtering and PCA and UMAPs, but it also leads to new kinds of workflows, because anytime we've seen scale in any previous modality in the history of AI and machine learning, it's given us this Sutton's bitter lesson, which is you can throw a bunch of compute and machine learning at large data sets and you can get a new paradigm of capability, right? So just some history, like ImageNet and AlexNet 2012 led to the computer vision era, and Common Crawl and GPT-2 unlocked the LLM era, and in bioinformatics, PDB and AlphaFold gave us the protein generative protein design era, right? So similarly, single-cell transcriptomics, single-cell foundation models are giving us the virtual cell era. And to get right into the data itself, as many of you know here, single-cell genomics, you store, transform, and compute with sparse matrices. So cells by genes, most of them are zeros, but at the scale of 100 million to a billion cells, you're looking at something like 100 billion to a trillion non-negatives, right? So that's a lot of data to fit on a single node, and the status quo paradigm has been to store in an HDF5 based file format called H5AD. The in-memory format is called ANData, which kind of gives us some metadata schema around the counts, the sparse count matrix. And then most of the computation is SciPy sparse, and we'll see some of the issues with that. So let's take a typical batch pipeline. I thought I would kind of like draw out the steps and talk us through some of the challenges that we have with this traditional H5AD and data SciPy sparse paradigm. Take a data set like a collection of H5ADs, much like Arc Institute's SC base count or cell by gene or anything like that. You typically have data distributed in a public storage bucket. The moment you copy that over to your private bucket for like your institution or your team's analysis, the distributor of those data sets is now incurring some egress cost. And it's not too far to hazard a guess that most of these downloads are going to result in like really simple computations, right? Most people are going to look up something very, very specific to on a very narrow subset of the data set that they download. So that's something to keep in mind. Then when you run some containerized workload, like whatever standard single-cell transcriptomics pipeline, you're copying that over to local SSD storage or network storage. And so like you're network limited there from your private bucket. Then when you're doing computation, you're IO limited by the particular nature of H5Py, which is like the Python library that reads HDF files, and that is limited by the GIL. In fact, this is something that the creator of ZAR noticed about a decade ago when he was, so ZAR is another cloud native format for imaging and climate data and so on. But interestingly, it came out of the genomics world where somebody at a research lab said, I have to manage 20 billion numbers in memory, and I can't do it with H5Py. So that's kind of how that happened. But it's interesting that H5Py is still GIL limited. So if your CPU has 8 cores or 16 cores or whatever, you're only using one at a time during I/O. Then of course, as Kenny mentioned in the beginning, you're approaching a multi-terabyte scale for these count matrices at the 100 million scale, and you're RAM limited. So because it's not cloud native in the sense that you can stream only what you need into memory and compute, you're now forced to vertically scale your compute. So you have to allocate bigger and bigger instances. And the way this might play out in a typical lab is you walk up to your cloud administrator and say, hey, I need more compute. And that can be a human in the loop that can depending on your institution, can take time, right? Then the other thing to notice is SciPy sparse itself is single threaded. So the sparse data itself is typically SciPy sparse format. Under the hood can be COO or CSC typically, but that's single threaded as well. So any compute you're doing, again, is limited there. And then the last thing to note is because of I/O, when you're writing things back to AND data or H580, again, you're I/O limited. Of course, writes are rare in this domain, but just something to keep in mind, right? So I mentioned this. It's kind of like not cloud native. There are human bottlenecks, and there's data duplication as a consequence of this. So if you're a team of 10 people looking at the same data set, you end up keeping 10 copies. One of that is just like the trivial consequence of that is like a 500 gigabyte data set ends up becoming 5 terabytes with 10 people. But the more insidious issue is just as you modify these data sets, you don't have lineage, you don't have metadata tracking, etc. So that's something to keep in mind. And of course, the community is aware of this, and what has resulted as a consequence of that is some form of coping, right? So you have different requirements on top of these data sets. So people want to visualize UMaps, and so they export the UMAP coordinates into Parquet or some other format, and then stream that into a browser. Likewise, for analysis directly from the cloud, they might use something like TileDB Soma, which is like another standard around cloud native streaming, and so on. So there's fragmentation in the data formats. So when I looked at this about 6 months ago, I sort of went back to the drawing board and said, could there be a better way in which we imagine a store once query in place format to support all these different workloads? Researchers are doing things in a notebook around a small subset of files that they sample, or an agent is executing LLM generated code in a sandbox, answering an LLM generated prompt with commodity compute, right? Visualization app just needs to stream that particular subset of cell types that you care about in that viewport, etc. You're doing multi-node training. Can you stream randomized batches to your GPU just in time? And of course, your traditional batch pipelines, right? So if you could do this, and like I sort of like give away the technology choices a little bit here, but if you could do this, you could save massively on egress costs, because now you're sort of incurring the type 2 cost as opposed to the type 1 cost, which is like copying everything. You just issuing GET requests and range requests and so on to parts of the data on a streamable format. And then of course, by escaping the GIL with like traditional H5Py, you get true concurrency with I/O, true multi-threading. You can execute things on commodity RAM and GPU as opposed to like massively over provisioning and underutilizing, etc. And you can scale your compute horizontally as opposed to vertically for like all of these workloads with a very high concurrency. I mean, modern file formats that will get into can do like tens of thousands of concurrent reads at a time. So there's plenty of potential benefits. One more thing to keep in mind is you also want to look at who are the consumers. I mentioned the workloads a little bit, but just to kind of give you a sense, like computational biologists care about exploring data without downloading it. A data engineer, say, who's distributing like a massive data set cares about egress costs and so on. Like a machine learning researcher wants to do really efficient training by allocating just the GPU they need and actually utilizing it during train time. The interesting thing to note is there's super diverse performance needs. I won't go through this big table, but you need everything from sub-second query latencies on like tens of millions of cells on commodity compute. You need to be able to saturate GPU cluster from a cloud store, and then you need like low latency queries of things like embeddings, because just like in other modalities, you have foundation models coming online and then embeddings of foundation models on top of which people want to do downstream computation, things like nearest neighbor search, etc. And so you need to be able to support all of those low latency embedding queries, nearest neighbor searches as well, right? So keeping those requirements in mind, this is kind of how I architected the stack. I'm going to break it down into things that you can mix and match. First, like, where do you want to put the data? Local or cloud, and if cloud, is it like a network file system? Is it object store, etc. Right? The next thing is the file format itself, and you have a handful of options for like cloud native table formats that I'll get into in a second about like the particular features and the bake-offs between them. The next layer on top of that is like, once you have the file format, can you independently choose a query engine, something that can read just what you need from those formats, and that you can sort of like mix and match for your different workloads depending on their unique capabilities. And then of course all of this is like the internals, right? But like keeping in mind the end users, computational biologists would still like to treat this new format as though it is AND-data, as though it is ScanPy, to be able to like slice a matrix or call from scipy.preprocessing import normalize, and then just call a normalize function, just do functional programming in notebooks without caring about the internal. So what are the right interfaces for folks like that? For machine learning training, you want to have like a really efficient data loader that's like shard aware and streaming and so on, but it should look and feel like a PyTorch training loop. So what's the right interface there? So I thought about like these as the categories which are like self-contained, within which you can make certain technology choices. As for storage infrastructure, I won't super get into the details, because I think a lot of these table and file formats are like compatible with most of them. But one thing that I just wanted to highlight is Hugging Face compatibility. I ended up choosing Lance, and I'll get into that next. But like, Hugging Face compatibility is great because like you get a human readable data sets repository, and you can have metadata in that repository. So you can do something like this, where Hugging Face in the past couple of quarters has pushed really hard on this HF API, and so you can actually point your data set to a Hugging Face repo, and then like write SQL and get some data back from Hugging Face. So far I've uploaded a few popular data sets like the Tahoe 100M and Parse Biosciences PBMC 10 million data set. And if you have a moment, I'll show you a demo about how you can query parse 10 million from an agent. And so it's important that I wanted to choose a file format that supported something like ease of metadata visualization and like human readability, as opposed to like, okay, here's a bucket, and also saved me building a bunch of front end for people to know what tools they could access. Next comes the table format. I talked about a few different ones, but I ended up choosing Lance for a lot of reasons. I think it's free open source multimodal table format. It's one of the few, so Lance is next generation to Parquet, and it's about 100 times faster than Parquet for random access, which is great for machine learning workloads because you want to pull random samples at different points in time. It's also multimodal, so you can put things like video or binary blobs or audio right next to your metadata in the same table. And it's also vector native, so you can put embeddings of those objects in future pipelines. You can add columns, and then you can append it with like multiple embedding vector columns. And then it has implemented things like vector database algorithms like HNSW, etc., under the hood. So your vector search is blazing fast up to 1 billion rows or so. You can get like good sub-second query results. It's got a number of other features like ACID compliance. So unlike a file format, it's more iceberg-like. It's a table format where you can do things like rollback and schema evolution and so on. So that's super powerful if you want to take a OBS column in a single cell data set and then enrich it with like a bunch of lookups or a bunch of compute later and add more metadata columns to it. I haven't seen another format that gives you that kind of flexibility for like schema evolution and time travel. And of course, they're really sort of like winning the multimodal lakehouse wars. They're doing great. They're adopted inside big tech now, and like Uber and Netflix and Meta all contribute to the open source format, etc. So it's a meaningful bet to make. Some more details. I think I mentioned a few of these, but when you really get down to a few of the details, like can you do separation of concerns? Like Lance is one of those few formats which talks to DuckDB and Data Fusion and like other and Polars and other query formats. And then it's really cool for stuff like schema evolution. They've really built in like versioning without making a 100x copy, like an identical copy of the data set. Whereas any other format, including TileDB, which is popular in this domain, you have to make a 100x copy just to add one more column. So I think for reasons like that, Lance is really great. As for the query engine that does things like push down filtering and so on, complex joins on multiple table formats, etc., on top of Lance, most of the contenders are like really awesome. But I ended up going with Polars, one, because I think the nature of compute in single cell is very much like data frame, and Polar is like Rust data frame, so you can do like data frame native expression of what you get out of those formats. But also among the formats, Polars is the one that's kind of leading the GPU acceleration of the query performance itself. So like you can execute Polars on GPU and accelerate your data frame workloads. But any of the others are like perfectly fine for a majority of workloads. Okay, so the SLAF schema itself is basically a collection of Lance tables that mirrors the AND data format. So for users of AND data, it should feel feature complete. So you have the expression data, that's a sparse matrix, expression.lance. Cells, the obs and the obsm, which are vectors, they both go into a cells.lance table. Likewise, var and varm go into a genes.lance table. And then most recently, I also implemented the varp and the obsp. These are like sparse pairwise data, which is, I think, potentially useful for spatial formats as well, right? So and then unstructured is just a JSON for now. But in principle, it's feature complete with the AND data format. So you could potentially start, taking a full AND data schema and converting it. I mentioned this briefly earlier as the top level of the interface, but if you're coming from ScanPy or AND data, you can just do that, read SLAF, and you get a lazy AND data object. This isn't actually data in memory, it's just a pointer to the SLAF arrays. And then when you do things like this, slice, it's also lazy. So it accumulates those operations. And just like in, if you're familiar with Dask, if you say .compute, that's the time when it actually sequentially executes these operations. Likewise, this is not feature complete with ScanPy yet, but for the most basic things like log1p and highly variable genes and so on, you have equivalent operations which are actually Polar's expressions pushed down to Lance. That's for the interface around ScanPy and AND data. I want to say a few things about the data loading as well, because I think this is an important issue if you don't want to waste the GPU that you've provisioned and stood up during your training job. Training jobs can last hours or days, and if you're underutilizing it because your data loader is blocking, that is, you're waiting for data to arrive from your storage into your GPU tensor, your Torch tensor, then you're effectively wasting cloud compute. And so because of all those things that you get from LANs, let me make sure. Because of all of the concurrency and like GIL issues that you sidestep because of LANs, the native throughput is pretty amazing. It's more than sufficient for training something like a single cell SCGPT on a single 8 times H100 node. There's a bunch of ideas that have gone into the data loaders. I'll just mention them briefly. One is thread-based. The fact that PyTorch scales with processes actually is a limitation. And in this case, I chose thread-based. Later discovered that NVIDIA, Meta, and Ray have also implemented thread-based data loading. So there's a lot of precedent there. There's definitely good content out there about the trade-offs between processes and threads. The second is I migrated all the transforms to an async prefetcher. So this is common. This is common because inside a training loop, you're saying for batch and data loader, and you're doing some compute. You're actually doing a bunch of things that aren't necessarily GPU native and better off for CPUs. In our case, it's things like self-filtering or like tokenization and so on. Those scale much better when you do it across many cells, and you can just do vectorized ops and polars. So that actually saves a bunch of compute as well. The third idea is how to make it completely randomized, how to have like fully random batches arrive into your data loader without pre-shuffling your data. This is kind of an important issue that a lot of teams deal with. But under the hood, what I've done is basically once you have sufficiently small number of shards, then your prefetching batch can just choose shards at random in each prefetch batch, and then you bring all of those, like you bring a sufficient number of cells per stream, per read, and shuffle them in memory. So there's a lot of stuff going on under the hood to make them arrive at random to your GPU without pre-shuffling. And then finally, because I mentioned this, the outer loop in data loading is very CPU bound. The inner loop is GPU bound, like your actual grading, compute, weight update, all of those steps are really GPU bound. So if you could break out your data loading transformation into CPU workers and then just have tokenized batches arrive to your GPU into your inner loop, then you have the possibility to utilize your GPU much better and actually independently auto scale those. So there's a couple of precedents there. There's a nice paper from Alibaba and then another academic group. So basically like bring the ideas of Kafka and streaming into machine learning data loading training. So I think a bunch of the ideas went into data loaders to make the throughput what it is. In the interest of time, let me skip ahead. I'm just showing you that conversion is super fast. There's CLIs and APIs that you can convert. And then there's a bunch of metrics on the docs page themselves telling you how much faster metadata queries are and submatrix queries are and so on. All right. I had prepared a couple of demos, but let me see how much time we have, and I can definitely tell you about how I have an SCGPT training run on Modal. Mr. Yes, I can take questions and I can show you. Actually, I have like an agent that just executed a query from the parse 10M. So instead of running that, let me just show you like what it just did. Okay, so this is basically something like Cloud Code, but like I built a web UI around that, and you can go add a skill. So I added a SLAF skill to just tell it, okay, you can go query Hugging Face like this, and like here's. Oh, okay, sorry. Sorry. Yeah, this is a web UI around something like a thin wrapper around Cloud Code, really. But you can have you can go upload skills. So I gave it a SLAF skill, and then I told it, well, this is how you find a data set on Hugging Face, and here's some example code for you to stream it. And then I mentioned that skill, and I said, analyze, parse 10 million cytokine data hosted on Hugging Face and answer the following questions, a lot of which are like metadata queries. And then it did some work, spent some tokens, and then gave me an answer to each of those questions, which is basically writing SQL and querying LANs, which is hosted on Hugging Face. Sorry, let me pause and see if there are questions, and then I can switch to the Modal demo if we have a second. Yeah, good question. I think the question is, how do you envision the future with respect to other modalities beyond single-cell transcriptomics? So far, I haven't thought about it, to be honest. But I think wherever with spatial transcriptomics, there's a straightforward step because there's H580 base formats, and so like wherever you have a cloud-native underlying format which you can transform into something as generic as LANs, there's an opportunity for SLAF to contribute.
It's kind of a naive question, but when you lazily load the data at the beginning, you kind of pay for that compute later, especially when most of the operations still require using all of the data, like the whole matrix. So in practical workloads, where do you actually see time savings, and what's the reason for it?
Yeah, I think that's a great question. The question is around how much upfront cost or how much cost downstream do you incur by making your computational graph lazy. I think the biggest win there is like for really small slices. Like if you're doing exploratory analysis and you're super used to an AND data interface, and then you want to say, hey, give me just like this rare cell type and like sample 100 cells, then your lazy accumulation of compute followed by like triggering the actual computation, that can save you a lot of money because you do things like push down filtering, right? Like you can push down a column and row-based filtering down to the query engine. But for batch compute, it's probably not going to buy you anything. So, but the way I've implemented batch compute in practice is to actually like break it down into chunks.
Yeah, I imagine you get a lot of time savings from the Lance format when you compare it to how like log1p or these operations work in naive AND data. I'm sure you've done benchmarking there.
Yes, but I think most of it is actually coming from the query engine itself. So like I think DuckDB, for example, has like very efficient log transform implementations and things like that. So Lance is just like the storage layer, and then these query engines are deciding like how to push down, how to optimize the SQL query itself and push it down. So a lot of the things like log1p, for example, are just written in SQL.
Cool. Yeah, we've been watching this project from afar. It's really cool and going to be even in more need as the single-cell spatial experiments scale up further. Nice segue to, as you scale up these experiments, how do you think about building foundational models to model transcriptional state? Dhruv here is going to give a talk on that.
Okay, hi everyone, I'm Dhruv. I'm a student at UC Berkeley, and I also work at the Arc Institute, and I've been training models such as state, stack, and I'm going to talk a little bit about them. My coworker Abhi did a presentation on stack here a few months ago, and I'll be going into some of like the downstream applications that we've used. We talked about state a few months ago, and I'm going to talk a little bit about the downstream applications of state, and stack is a new model we released about a month ago. Yeah. So yeah, I'll be talking about state and moving towards large-scale simulation, stack, a little bit about the inductive biases in the model architecture, and really trying to scalably simulate perturbations, and also going beyond. And this will be more of a machine learning talk,
And hopefully I'll have some interesting takes, and the views of these presentations are my own and not of the Arc Institute. So let me contextualize the entire problem we're trying to solve to begin with. Kind of what Noam was talking about earlier, we're scaling up these single-cell transcriptomics readouts, and we're doing this thing called perturb-seq, where we perturb these cells in some certain way compared to their baseline state. And we get these transcriptomic readouts, which are basically just like a histogram of all the mRNA counts in a cell. And this is a really interesting problem space because you could take something like a disease cell and a healthy cell and look at the differences between them. And a real virtual cell takes in input of a disease cell and predict which perturbation would lead it towards some sort of healthy state. So this is kind of just a long-standing goal of in silico modeling of biology, and this is the problem we're trying to tackle. So first I'm going to be talking about state, paper released earlier this summer. So the state transition model learns perturbation effects on covariant matched sets of cells. So it takes in these sets of cells with the same perturbation at the same time. It's a basic transformer. It's bidirectional. We don't have a tokenizer. And we learn to predict perturbations across these big sets. So the exact learning problem is actually looks very similar to language modeling. Like you have the same batch size, your sequence length, and your dimensionality, which is just the number of genes we're measuring. We use HVGs here. We use like the top 2,000 highly variable genes. I'll talk a little bit about this when we go into stack and why that design choice might be problematic sometimes. And our loss is simply just like an energy distance MMD computed with this sort of kernel. And yeah, so we have our inputs of transcriptomic readouts of control cells. We indicate which perturbation we're doing, and our output is these predicted transcriptomic readouts post-perturbation, and we compare it to our actual data. Something really important is that when you do single-cell sequencing, you're not sequencing the same cell pre- and post-perturbation. Sequencing kills the cell. So you're learning over a distribution here, which is why we need this energy distance MMD. And what we see is that state actually scales with cell set size. When we have more cells in the set, and when we train for longer, we actually see decreases in validation loss and increased performance. And this model, indicated in the blue, is really state-of-the-art on a lot of metrics. If you're interested in this, you should check out the paper or Ubbi's talk, which I think was recorded previously. Yeah, so state is really state-of-the-art, beats all the baseline, beats every model out there, and it really does really well with bigger data sets. As you see, the largest data set we tested on Tahoe, we have huge boosts over baselines. And today I'm going to talk a little bit about going past metrics, UMAPs, gene set analysis. People do a lot of these analyses for real single-cell data sets, but I think we can do something a little more interesting because we actually trained a machine learning model and has a very developed feature space. So can we learn anything about that feature space with cool out-of-distribution experiments? So this is just a nice figure from this paper in goal-conditioned RL where they take states and they represent them as a dual representation, which is just the relationship from other states. And assuming we have a developed feature space in the state model, can we learn something about super out-of-distribution in silico predictions, so like really weird perturbations, just by comparing them to other perturbations that the model predicts? So what this really looks like is you just kind of have these cells and you have these different perturbations, and we're just going to represent them as their differences between other perturbations. So this is one of the first experiments I tried, was we took the state model and passed in all the perturbations that we have from the Tahoe data set. And these are just like a ton of different drug perturbations with different dosages. And then we took those outputs from the model, and then we passed them back in and did another combinatorial space search over all the other drugs. And the Tahoe data set never did any combinatorial perturb-seq experiments, but now we have these sort of like drug plus drug perturbations predicted from the state model. And then we also have all the single drug perturbations, and we could just add them together. So we have these deltas from control cell, and each one of these perturbations, we could just add up each combinatorial pair for each single perturbation, and then we also have these like model predicted drug plus drug perturbations. And we learned something really interesting here. Like purely trained on these single perturbation drug data sets from Tahoe, we find that the model predicts these non-additive effects. So what you see here is basically if we add two drug effects from the initial single perturbations together, from delta from control, we get some sort of effect. And when we pass the cells in twice within the model, we have some sort of different effect. And we take the delta between those last two effects. Sorry, there's a lot of deltas. And we find that like these combinations with the drug abiraterone are most non-additive. And we really wanted to verify if what the model predicted was correct. So we looked at this data set called DrugComb, which actually predicts the drug, it's real data of drug-drug interactions in the exact same cell type in the Tahoe data set. So we don't have single cell sequencing data of drug-drug interactions in the Tahoe data set, but our model predicted that these drugs with abiraterone would have weird non-additive effects. And we actually fit the data pretty well. And across these like metrics that DrugComb has, like the Bliss score and Zip score, which basically just predicts synergy, we're actually predicting this non-additive effect. And the model was only trained on single perturbations. So this is a really interesting form of generalization. And we try to push it even more. So we try to approximate genetic perturbations through combinations of drug perturbations. So what we do here is we use another data set, Replogle, which has a couple cell lines overlapped with Tahoe. And we also use a data set from Milia, which is this biotechnology company. And we take their genetic perturbations, which are just CRISPR-I knockouts, and average them over a ton of cell lines. And then we look at the drug perturbations in the Tahoe data set and compare those deltas. And we take those big deltas of those genetic perturbations and express them as a linear combination of 20 different drug perturbations. And what's really nice is that the way that state architectures works is that each perturbation is just one hot encoded. So this linear combination is just like making it less than one hot, and we can just take a ton of these like one hot encodings and scale them up or down, and we get this like nice linear combination. And what we find is that we can do this super large scale simulation. We have hundreds of terabytes of simulated genetic perturbations on these Tahoe cell lines. And we see very interesting sensitivity to certain genetic perturbations in the 50 cell lines of Tahoe. This is just, I think, one cell line. We see like that the KRAS G12C mutation clusters together. We see a lot of interesting canonical similarities and also non-canonical similarities, which is just basically showing that state as a model can be used for really interesting things just past normal single cell analyses. I'm going to move on past state now, and I'm going to talk a little bit more about the machine learning. So there was this great talk from this OpenAI researcher who talked about structure in machine learning and what really makes machine learning models work. If you think about earlier when Attention is All You Need was released, the transformer architecture really looked like an encoder and a decoder. And over time, when GPT was released, we've started moving away from the encoder architecture, and we just have a pure decoder. And if you think about this, the shift is really more about just removing structure in the model. And these inductive biases that you place into the model are largely controlling what you can learn from the data. So when you have more compute and data, you basically want to remove more structure from your model in order to let it learn its own internal representations instead of forcing these structured representations. And when we look at the state architecture, there are a couple of design decisions that are not really optimal. So when we look at these two encoders in the beginning of the architecture, the basal encoder and the perturbation encoder, we control a lot of things. First, we control the amount of HEGs per data set going into the basal encoder. And this basically makes it so we could only train on one data set at a time. If we train on Tahoe, we can't train on another data set. And if we do that, we have to throw out the encoder and then fine tune a new encoder for the new data set. And this really just like limits our learnability. We limit ourselves to a certain amount of genes, and it's really not optimal. And you see the same thing with the perturbation encoder. Since we have these one-hot representations of perturbations, we're stuck with those perturbations, and we have to do those really hacky things I talked about earlier, like passing things through a model multiple times to learn a new perturbation. And if we really want to just truly scale this sort of like in silico simulation, we need models that are not trained per data set and can continually be learning from new data being brought in. So now I'm going to move on to Stack. This is a great project led by a PhD student at Yale, Mingze, who was also at the ARC at the same time as me. And this model really focuses on breaking out those, putting the right inductive biases in a machine learning model to just like really in context learn a lot of these big perturb-seq data sets and non-perturb-seq data sets as well. So going from HVGs to sparse gene padding and modules was a big part of this model. We, instead of having these sort of like 2,000 size vectors for each input cell, what we do is have these sort of like sparse matrices where we store lots of 0 values for lots of different cells, but we have a huge gene space. But we really don't want to like input this huge gene space into the model because having 95% zeros is not really going to let your model learn much. Yeah, you're not going to really get that great gradients because you don't want to have your gradients keep on hitting zeros. And so what we do is we down project this into gene module space. And this gene module space is about 100 gene modules. And they basically, this is learned over the time of training, and they basically just encode gene ontology terms. We'd have no limits on what the model can learn. And when we just look at what the model learned, it literally just encodes all these different biological processes, which is a pretty neat result. So the architecture in state, we had this like set style framing where we have a set of cells and we do attention between them. And then we have a loss, MMD loss. In stack, we also do attention between sets of cells, but we take it one step further. We do tabular attention where you can learn within a cell and between cells. So tabular attention became really famous in the last year from TabPFN, which is a great model. You guys should read the paper if you haven't. Yeah, I'm not gonna go too deep into this, but it really just allows for more learning within a cell as well. So with these gene modules, we do attention between cells and within themselves. And this results in like really nice embeddings for each one of the cells. And these embeddings are compared with a reconstruction loss. And a really big part of this was actually systems engineering on the data loader. We did not use SLAF, but we kind of hard coded a lot of this like data loader work, which was really important. And we had to get the MFU up if we really wanted to iterate on model architecture and a lot of things. And we trained on a ton of human cells. A lot of this is from SC base count. And yeah, the way that we actually learn things is that we randomly mask things out in these chunks, but in these like gene by cells matrices. And the model tries to reconstruct those random maskings. This is kind of similar to diffusion language models. I'll talk a little bit about that later. And yeah, this works. The right covariates, scaling model size, scaling context size. It improves reconstruction loss over training. And also at test time, we have this plot in the paper where we increase the amount of unique cells in the cell set. And at test time, we can see better performance, which is great. And yeah, it's really state of the art. It was trained on like less than 150 single cells, and it's incredible at these classification tasks. And a lot of the training data was actually immune cells. And you see in like these lymph node atlases, we are so far past state of the art, which makes me really bullish on what these models can learn in the future. Because if you have these really big data sets for lots of different sort of cell types, there's a lot to go. And Stack was also the first model that actually used post-training. And this is one of the really exciting parts about this project. We use self-distillation from the Stack pre-trained base model itself to learn these interesting prompt query adaptations. And the way that we do this is that we take some sort of perturb cells, and we also take control cells. And from the perturb cells, we're able to just like swap them out with these control cells and allow the model to learn to change those student cells into the teacher embeddings. And this is really interesting because self-distillation actually in the past month became a really big deal for RL on language models. And so self-distillation is a really interesting idea. And yeah, if you guys want to talk a little bit more about this, come talk to me after. But yeah, now we have a post-trained model that can basically convert any control cells into perturb cells just by looking at the other perturb cells in its prompt. This really goes past that previous idea we talked about, the perturbation encoder being really limiting, because we have to express different perturbations and like hard code them into the encoder. Now we can literally just learn in context. We put a perturbed cell in from a different cell type, a different donor, and we could put a different control cell in from a different donor, and we can just learn that effect and pass it over into that new control cell. And this is just allows a ton of new applications. And in the paper, we apply this post-trained model on a huge data set, and we release it it's called Perturb Sapiens. So Perturb Sapiens spans across 28 tissues, 40 cell classes, and 892 different perturbations. It is also immune cell focused. It's a PBMC perturbation data set, and it's really tissue balanced. We have a ton of different drugs and cytokines, lots of different cell types, and we released it for the open source so a lot of people can hack around with this data and see the differences between model predicted perturbations and big single cell screens, because you're really not going to get single cell sequencing screens at this scale anytime soon across these like tissues and have like the same batch effects across the data sets. And Stack also, just to make sure that this data set makes sense, it generates functionally similar outputs to the real data. I think this is a bit small, but the main thing you should be seeing here is that these first 3 rows are real data, and the first 3 rows in the Stack generated like basically match up with the real data. We're predicting basically the same things, and then we're able to extend it to all these other data sets, and we're predicting a lot of interesting things that are not shown in other cell types. So Stack is predicting interesting things, it's preserving the priors, and also look out for some upcoming wet lab validation, which will be really interesting with donor specificity in T cells. So now, I guess moving past state and Stack, I wanted to talk a little bit about where the field is going, and hopefully some mild takes on what people should be doing for really pushing perturbation modeling. So this is a great quote from Alec Radford around, I think around GPT-2, where it's pretty prescient, and he was talking about GPT-2, and he was like, if every time we train a model and we have our initial phase of creating a data set, basically everything that's not in the data set, you can't expect the model to know it. And he talks about our poor models, they know so little, and yet they have so much hidden from them. And this blog kind of talked about like the internet being like the biggest thing for language models. Without the internet, nothing would have worked. And in some sense, language had it easy in that case. Biology has nothing like that. We have to pay for our own data. We have to create this data. So let's think about what should that data actually be. So single cell data, perturbation data is rapidly growing. Tahoe 100 million was released earlier in 2025. ArcBioHub Tahoe is an announced data set that is being created right now. And SC base count, they had an updated preprint with now 502 million cells, so you guys should check that out. But I would say that the single cell space is not growing rapidly enough. This is a plot that I kind of put together with assuming chinchilla S scaling, which is earlier scaling laws done for language models. So I take a lot of the data points from Epoch AI, where we assume that flops per dollar is dropping about 40% every single year, and we assume Stack's amount of flops per cell. The amount of cells we start off with is just the SC base count paper, so 500 million. Single cell sequencing dropping about Muir's law. And if you have like a reasonable yearly budget, it basically shows that you should be spending everything on data. This y-axis is the percent of budget on data, and it accumulates over years. Basically, for the next 5 years, you should just be spending almost all your budget on data. Obviously, you need like a development cluster, and you need employees. But if you're just doing this sort of like data versus compute allocation, you should be spending a lot on data. And obviously, the question that comes after that is what is the right data to be scaling? I think a lot of startups are tackling this question. And yeah, there's like the three main axes I think about are biological system, perturbation, and assay development. And these all change a lot of different things. If you change the biological system, it changes the downstream application. As we saw earlier, Stack was trained on a lot of immune cells. It does really well on immune data sets. If you change the perturbation, it changes the model you build. If you're doing a lot of time delimited perturbations, you're going to train a generative model for sure. And assay development obviously changes the systems you build. You just saw Cyto. If you're using 10x Flex and you change your guide system to be like sort of the pooling with 10x Flex, you're going to have to build systems to operate at that scale. But I would argue that the biological system part is the most important. And it is the most important thing to figure out in the upcoming future. And I just wanted to highlight this one, pretty much just a biology paper that showed that immortalized cell lines are not actually that bad as people always claim. If you do transcription factor perturbations on immortalized cell lines, you can recover a lot of the same clustering and atlases from in vivo studies. So this is a great paper from the Norman lab. And I really encourage people to look at all the immortalized cell lines people are doing Perturb-seq on, and making sure that those immortalized cell lines actually reflect the in vivo biology you care about. Because if you're training a model on these like huge Perturb-seq data sets, but they don't really generalize to in vivo, it basically just does not matter. If you want to do donor specificity, which is kind of the downstream application of a true virtual cell, you need models that can actually generalize to in vivo, and that means you need data that can generalize to in vivo because your loss is from the real data. So yeah, someone really needs to go deep into every single immortalized cell line that we use, and preferably not Claude Code because it just thinks I'm a genius biologist. And yeah, if you want to see if there will be scaling, you need good tools like Cyto and model architectures like Stack. This is just a plot from the Nichilla paper. We're still up here. We want to go down here and see if scaling actually holds for perturbation models. And I think that's it. Yeah, just some acknowledgements. Yusuf, my manager, and leads the ML group at ARC. Abi, who's been kind of a great mentor. And Dave, the CTO at ARC, and Mingze, who led the Stack paper. And obviously the rest of the ARC team, including Noam. And yeah, feel free to reach out. Any questions?
I have a couple of general questions. On your first slide with the problem statement where you said, well, essentially we want to predict the set of perturbations that lead from disease cell to the healthy cell. The definition of a healthy cell, is it based on gene expression or based on phenotypic morphology in your problem statement?
Yeah, so everything is in transcriptional state. So you take control cells from a healthy patient and then you sequence all the mRNA. And that's your definition of a healthy cell. Obviously, you can do bigger multimodal things where you integrate tons of different data types together, but the models that we've been training is just you take a healthy cell and you get the mRNA out.
So then the question is like, to reliably train your model, did I get it correct that you really need to show the training data, all possible combinatorial combinations of perturbations, because I assume there's a huge high-dimensional space for you to reliably find the perturbation, which is kind of an optimal parameter that will lead to the health.
Yeah, so if you have 20,000 protein coding genes and you want to explore combinatorial space, you cannot order all those single-cell screens. No matter how much money you have, you cannot do that. And there's a lot of proofs of concept that combinatorial space is very interesting. Yamanaka factors are 4 genes, 4 transcription factors, and so there's lots of proof of concepts that like multiple genes are very interesting to work with, and this space doesn't even only look at gene perturbations. First of all, for every gene perturbation, you could do CRISPR-I or CRISPR-A. You could upregulate genes as well, and the Tahoe data set is actually just drug perturbations. The drug space is completely gigantic. In this stack, Perturb sapiens, a lot of the perturbations are actually just cytokine signalings. So the perturbation space is just like absolutely insane. It's super big. you could assume that the 20,000 protein coding genes, those single perturbations, probably in the next 3 years, most immortalized cell lines will have data sets that do like a lot of the most interesting ones in there. But the combinatorial space will never be explored just by humans, which is why we need those models.
Thank you.
Yeah.
I'm curious if you've done any work on like interpretability of these models and seeing, are they like developing some sort of like biologically coherent, like mechanistic understanding of how these perturbations like lead to their effects?
Yeah, so in the original state paper, we have a supplementary figure on interpretability of the models, and we see that the model sort of encodes these like pseudo-encodes certain baselines like perturb mean, but then also learns a lot of like interesting cell-to-cell effects. Yeah, so I would look at the state paper's supplemental for that. Yeah?
I want to ask about the chinchilla scaling laws that you talked about. You mentioned something like 20 tokens per param is like considered like a, I guess like a default of the group. Don't you think in single cell we're like much larger than that? But at least like, I don't know, right tokenization schemes, you know, if you're training 100 million cells, 2,000 tokens per cell, we're like way over that, right, relative to our models.
Yeah, I did not assume 20 tokens per cell.
Or per param.
Yeah, so 20 tokens per parameter is straight from the chinchilla paper. Like when they trained the Gopher models, they got that. But for that plot, I used kind of like a conversion there. So basically for each vector that went in, I got like the flops that actually went to that vector. And then I converted it to stacks flops per cell because each cell is just a vector. So it was like kind of like it was like a flop by flop equivalence rather than tokens, because we don't use tokens because our cells are too stochastic.
How far do you think we are from that, getting to that ratio? Because right now we're...
Yeah, I mean the smallest models in the chinchilla scaling laws are basically the sizes of our largest models around right now. So I'd say we have a lot to go. Yeah.
For the combinatorial predictions that you make in state, obviously talking about distributions of transcriptomes or transcripts is like a little suspect. You can use like UMAP or something. But I wanted to ask for the drug pairs where you found the most interesting effect, how far in or out of distribution were they with the company data, or with the starting data?
Yeah, so these are pretty far out of distribution. The model has never trained on its own outputs, so it doesn't have basically when you pass in its own outputs into itself, the model kind of breaks down a little bit. And like you would not get, if you got like real, with the same batch effects, if you did the Tahoe perturbations with multiple perturbations, you would not get much synergy between that model double predicted perturbation and the real data. But the point of that plot was kind of showing that like if you compare it to all the other perturbations that the model predicts, they're broadly coherent, which is showing that like the model is learning something like a world model. I wouldn't actually say that yet because it needs to be proven a lot more. But the idea is that you're learning a general perturbation space just from training on singular perturbations, which is sort of just a proof of concept. Yeah.
Okay, we gotta.
Yeah, no worries. You can talk to me after.
Yeah, come find them afterwards. Yeah, I mean, there's like a million questions I'm sure everyone has, so definitely find Dhruv post. Last, we got Ben from Modal, and he's going to talk about serverless computing for computational biology.
All right, so before we got here, I made a prediction that I would have very little time to talk, but I would spin that to my advantage and your advantage and keep my talk short. And I was going to even just use like TL draw and kind of like whiteboard it, but I think I'm even going to skip that part. So I'm just going to talk to you, kind of like step back a little bit, introduce you to serverless computing if you're not familiar, and then map it, like explain why it maps really well to computational biology use cases. And I'll like kind of have like a little toy example that I got from my friend who does protein engineering, who I used to work with and uses Modal. I'm not going to talk too much about Modal per se. I'm just going to generalize it as serverless computing. But you can go look at, Modal, you can go look at our docs, you can go read about it there, you can come talk to me or other people at Modal afterwards if you want to know specifics. So let's start by answering a quick question, which is, what is serverless computing? So it's one of these weird terms where it's like there are servers We call it serverless because the user doesn't think about servers. So that means instead of thinking about, and actually this maps a lot to Max's first talk. Like in fact, I was thinking his whole early slides about Kubernetes could just be stolen and we should be using them at Modal. Modal is also built from the ground up to be a replacement for like Kubernetes, for example. And so the idea here is, as a user, you're just going to think about writing your code. So let's say you have some function that does inference, or it does some computationally heavy task. Your job is just to think about that function and then deploy it on Modal or another service. And then whenever that function is called to run, the platform is going to decide whether to scale up or down compute resources for you. All you need to do is tag a little bit of a description of what those compute resources should be on the function. And so like actually over here, you see a little decorator, maybe not, it says at app.function, and then parentheses GPU equals, and then you put A100, B200, whatever, and then you just decorate your Python function with that. And whenever you call that function from elsewhere in your code, we're going to run it in the cloud for you. And if you already have one running and there's only one call per container, let's say, and you call it again, we'll spin up another container for you. And then like all the, a lot of this stuff that was brought up initially is all solved as well. There's no YAML, scale from 0 and up and back down to 0. All that stuff is good. And like one of the key phrases in serverless computing is like pay as you go. So if you don't like have like some kind of consistent load 24/7, you don't have to pay for those resources 24/7, just when you ask for them. Right, and so that you as a developer, you think about things in terms of a function, not infrastructure. All right. Right, so yeah. And like Modal comes, yeah, so just to go over the list to match Max's talk. So there's no YAML, you scale to and from 0. We aggregate across all cloud providers, probably ones you haven't even heard of, across the whole globe. So like there's high availability. And this is especially good for Compbio, which is not like a latency sensitive use case in the sense of like how long is the take a photon to get around the world, which for some use cases is a really, really long time. But for Compbio, it's not. Like hundreds of milliseconds does not affect your overall like runtime. Like we saw in an earlier talk, things are measured in minutes, right, or hours, or days even. We have OTel and Datadog integration, dashboard, load balancing, all of that you don't think about. All right. So why does this work so well for Compbio use cases? And the reason is because like if you compare it to other, say like AI or ML, let's just say AI use cases, a lot of those, you have some single model. And the user just is going to call it and get back a single response from one forward pass. That's like the general state of most AI systems. But in Compbio, like one job is more like a giant call graph of different things. And inference might just be like, there may be like multiple models running inference. It's just one little step. And you may need to run that same inference over, say, like 1,000 different inputs. So it's a much more complex job that has a very dynamic shape. And that shape is often not even known till runtime. And so that's why being able to scale as you make function calls is like a really good match. The other reason is that each of these models or inference calls, let's say, might require a different GPU. And so you can attach a different GPU per function. Whereas if you have some like finite or like predefined set of compute resources, you just are stuck with what you have there. Whereas with a serverless platform, let's say, well, I'll get into some details on this. I'll give you specifics shortly. The other thing is, like I mentioned already, a lot of times your workloads are really bursty. So let's say you have a paper coming up and you need to run a bunch of experiments. So like even over like long scales of time, maybe you're like running a bunch of, you collect data for a few weeks, then you run a bunch of experiments for a week or two, and then you iterate on that. So you may have like long downtime on long time scales, and then within one job, you may say, spin up a bunch of GPUs, make a bunch of proposals or do whatever the inference for that model is. And then you have to evaluate all those on CPUs. Or maybe you want to send all those down into like some other model that uses, maybe it's like one large model that's running on a B200. So there's also on short time scales, this sort of like burstiness or variance in the shape of what infrastructure you need and on your load. All right, cool. And then lastly, this one's maybe obvious, is that like, let's say you have like the model we were just looking at, and you're trying to evaluate a bunch of perturbations on a cell type. Well, with serverless computing, you just run that in parallel, and it takes the same amount of time to run 1,000 of them as it does one. And when you make the call, like if you're using Modal, instead of just function call, you just do like .map, and that's it. And it'll spin up all the containers as necessary. All right. So let's real quick, we're going to take a specific use case and maybe like walk through it a little bit to make this a little more concrete. So the use case I'm going to have is like, let's say we want to design a promoter, and we want to have that promoter target like specific cell types in the retina, but not all of them, right? That's what like cell type specificity is all about. So you don't just want to say like how to do in this one cell type, but how to do across say like 10 cell types. All right, and so to do this, what do we have to do? So first, we need to propose sequences, and then we need to evaluate those sequences. Both of these steps are computationally intensive. So you're going to have probably one model that's going to generate proposals, and then you might have another model that might like evaluate chromatin accessibility or something like that, which will be like your stand-in, or like a metric, which gets you at the cell type specificity. But okay, yeah. So let's focus on the proposal one first. So for proposals, let's say you have some like really beefy model, and it does a really good job. Well, you might want to distill that model down, and then first use that model to make, say, like 10,000 proposals, and then run some evaluation, and then take that smaller set of proposals and use that to seed like a bigger model that maybe needs to run on a bigger GPU. And so you can kind of do this like iterative thing, and it's very simple from your point of view as a coder. And it really takes advantage of this like elasticity of the serverless computing pool. All right, so let's talk about evaluation, because this one gets a little more interesting. So let's say we're talking about like chromatin accessibility. So we have 10 cell types, right? So we need, for the use case I was talking with a friend about, so he has one model per cell type, or it's like a model fine-tuned per cell type. And so for each proposal, he needs to run 10 models. All right, so I guess you can kind of see how this like graph is like fanning out and fanning in. And for each proposal, we now have like 10 models to test, but it actually goes one further because he does an ensemble of models for each cell type. That helps him get like sort of an estimate of uncertainty and also some validation. So now for each proposal, let's say we generated 10,000, we have to run like, let's say there's 5 per ensemble, we have to run 50 inference calls. And so with serverless computing, you just do the first map, and then you do like another map, and it's very simple, and the compute pool will scale up and down for you. And this is, keep in mind, this is like as opposed to having like maybe a couple GPUs on-prem or some like reservation of like even like, let's say 20 nodes, the variance in GPUs is going to be low, and you maybe have a lot of people you're working with who are all converging on that same GPU or that same compute resource pool. Okay, cool. And then, yeah, so the last thing I'll say is like another application here is even for training. So a lot of times when you're training, you have to do many experiments. You have to search over hyperparameters. Or you may like, or like you may want to try different models or do fine tunings. So all of that, again, it's like very trivial. I mean, the kind of big takeaway here is that anything that's like trivially parallelizable, you sort of get that for free with serverless computing. All right. Yeah, and the last point I already made, which is that the other advantage is that you have access to very heterogeneous infrastructure. So you can, have different models of different size and really match the resources to the model appropriately. And so I guess I would say, if any of you are not using this kind of framework for your workloads and you would find it interesting, you can check out Modal's website. You can come talk to me. You can also check out LatchBio, which in a way is like very similar. Like they do differentiate at a certain point, but like that's one of the reasons that we're here together is that we both kind of see this as an important pattern as computational biology scales. So maybe, take a couple questions. Yeah.
So I should say first that I'm usually Modal evangelist, but I had actually a benchmark. I'm curious, when you started container, presumably there was like some startup cost.
Yeah.
Like across like most of the bio-workloads you see, how much does that like save people?
Yeah, so for cold start times, I can speak generally. I can't speak specifically for comp bio. But okay, so what I can say is at Modal, it's built from the ground up with cold start time in mind. So for a container, just to get it spun up with a GPU, let's say before you've loaded your model onto the GPU, that's like 3 seconds or less. So it's extremely fast. Like that is one of the things that Modal does differently from a lot of other serverless providers. However, if you have a very large model, it's still going to take time to load it onto the GPU. So another feature of Modal that's really nice is that we have GPU snapshots using the CUDA checkpoint library, which is not like that widely used, which means you load your model onto the GPU. You may even like warm it up, like warm up the cache, run a couple inference passes. There may be other things you're setting up on the CPU memory. You set your state all up, and then we'll snapshot the whole thing. We'll copy from the GPU RAM to the CPU RAM, copy all the RAM off, store it for you, and then the next time you spin up a container, we just load that state in. And so like for models like OpenFold or something, sometimes it can take a couple minutes to load it onto the GPU, and it goes down to say like sub 20 seconds. So that is something that we are optimized for, but it can be, can be a pain point of serverless computing. So there is like, like a lot of things, there are trade-offs. But I think for Comp Bio, I mean, in my opinion, it maps very nicely to serverless because these timescales are still like shorter than a lot of the workloads, Like that's when it gets bad, is when people are like, I only need to use this for 5 seconds, but it takes a minute to start up. Yeah. Any other questions? Okay, cool. Thank you very much.
Thanks. I know it's late. Please take pizza. We have extra, so if you want to grab a box, just do that. Thank you so much to Modal for co-sponsoring this with us. Huge, huge joy. We're probably going to be using them pretty soon with the project that Max described, because it's agnostic to kind of the container service we use to spin stuff up, so probably see synergy there. And then thank you to all the speakers. We'll record this, and the slides and stuff will be online. So thanks for coming. That was a lot of fun. We'll do it again soon. Thanks.